Hey sweetest potatoes. So in today's video we'll be studying about bZip and bHLH domain, which includes Leucine Zipper and helix loop helix. So the bZip and the bHLH domain, they combine two functions. One is the DNA binding and dimerization. And ZIP stands for Leucine Zipper and HLH means helix loop helix respectively, which are the dimerization motifs. Now coming to Leucine Zipper. So this Leucine Zipper motif, it mediates both the DNA binding as well as protein dimerization, and it is responsible for DNA binding. As you can see in the diagram, it is made up of two alpha helices, and they're joined together by a coiled coil, and these helices are held by the interaction between the hydrophobic amino acid, which extend from the two sides of each helix just beyond this dimerization, and they separate to form just beyond the dimerization they separate to form a Y-shaped structure, which will allow the side chain to contact the major groove of the DNA. And as you can see here, this dimer, it grips the DNA like how a cloth clip or a cloth spin, it will go and bind to the it will go and hold the cloth. So to better understand this analogy, so let me make this easier. So consider this thing to be your a cloth clip, and the green thing to be a DNA. So how the Leucine zipper exactly holds the DNA in this way. And now you guys can try it at home. And now coming to the different structure. So this is a structure of Leucine zipper, which is binding to the transcriptional factor GCN4, it's X-ray crystallographic image. And this is another image of Leucine zipper binding to the GCN4 DNA target, and A shows the side view, and you have the end view or the top view of the DNA in the second diagram. And these helix, this Leucine zipper also has a thing which is called as the heterodimerization. So what is this heterodimerization? So because of this heterodimerization, the Leucine zipper can alter their DNA binding specificity. So coming to heterodimerization, most gene regulatory proteins, they bind to DNA as homodimers. That is, they have two identical subunits. And the gene regulatory proteins can also bind to DNA with non-identical partners, that is referred to as heterodimers. That is, they have two different subunits. And these heterodimers, they typically form two proteins with distinct DNA binding specificity. Mixing and matching of DNA regulatory proteins in this way expands the DNA binding specificity as these proteins can display. So now you can see that in diagram 7.0, there are three distinct DNA binding specificity which is observed, which are generated from two types of Leucine zipper monomers. So here you can consider one monomer to be in dark green and the other monomer is in light green. But then it can form three distinct DNA specificity. And the dark green will go and bind to these regions which is in blue color, which refers to the sequences, and the light green will bind to the red sequences, DNA specific sequences. And when you have a heterodimer, so this is a heterodimer where you have a dark green and a light green coming together and binding to like the specific DNA regions. So similarly, if you have like three monomers, this is for two monomers. So if you have three monomers, then how many DNA specificity can you see? You can see about six different distinct DNA binding specificity can be seen from 3 monomers and so on. And these heterodimers, it is also an example of combinatorial control. So what does this combinatorial control? It refers to where there is a combination of proteins. So rather than individual proteins, when there is a need for a combination of proteins to control a cellular process, heterodimerization is important. Now coming to the functions of the Leucine zipper. So the bZip monomer, it consists of long alpha helix, and the C-terminal half in Leucine zipper and the N-terminal half will go and bind to the major groove of the DNA in a sequence-specific fashion. And in bZip proteins, nothing holds the DNA in place than the Leucine zipper. So the grip which is given by the Leucine zipper is so strong that the binding, it is better than all other DNA motifs. And examples include AP1 activator protein 1, and which includes many families like GenFos, activating transcription factor, MAF, and all of these, they form homodimers and heterodimers to Leucine zipper domains, and different dimer combination will recognize the sequences uh, and the promoter and the target regions. So now coming to the next one, which is the helix loop helix, which is abbreviated as HLH. So basically, these helix loop helix proteins are dimeric transcriptional factors which are found in almost all the eukaryotes, and they play very important role in embryonic development, which includes neurogenesis, myogenesis, heart development, and also hematopoiesis. And these are also involved in developmental process like sex determination and muscle development, etc. Now coming to the structure structure of the helix loop helix. So this helix loop helix, it relates to the Leucine zipper, and as you can see here in this diagram, the helix loop helix, it consists of two alpha helices. You can see here in the blue and the green ones, two alpha helices, and these are connected by a loop, and this loop is in red. And as you can see here, the alpha helices are not of the same size. You have a shorter alpha helix here, the top part, and then you have a blue one also, and a longer alpha helix, which is connected by a red loop. So in general, so you can see the second loop is longer. The second alpha helix is longer than the first one. So in general, one helix is smaller, and due to the flexibility of the loop, it allows the dimerization by folding and packing against the other helix. And this larger one, the larger helix, they contain the DNA binding region, and the two alpha helices, they extend from the dimerization interface, and they make a specific contact with the major groove of the DNA. So you see that the longer alpha helix will go and bind to the major groove, and the longer and the smaller helices are connected by the loop. And here you can see a few diagrams, which is the crystal structure of the complex of MyoD and its DNA target. So here the cylinders will represent the alpha helixes. So the basic region or the longer alpha helix is given in yellow, and you have the shorter alpha helix in red, and then the loop. So location is eukaryotic protein and the binding site is major groove. And then sometimes what happens is that in some of the products like oncogenic, that is cancer causing proteins like MYC as well as MAGs, you have you need the bHLH as well as zip domains. That is, they need both, not just the helix turn helix and the zip. So there is also a need of both the helix turn helix as well as the Leucine zipper, and it forms a domain which is called as the bHLH zip domain. So what happens is that, and the difference between this, just the bHLH as well as the bHLH zip domain, is that here you have the involvement of both the Leucine zipper as well as the helix loop helix, and so this type of interaction is needed when so that the monomers are held properly. So previously I told that the Leucine zippers will give a proper grip. So when there is like a lot of proteins involved, for example in these oncogenes, uh, just the helix loop is not sufficient to hold the DNA in place. So that is why they need a combination of both. It needs the help of the Leucine zipper. So the Leucine zipper along with the helix loop helix, together they will facilitate the interaction. So now coming to this to summarize. So the bZip proteins, they dimerize through Leucine zipper, which puts the adjacent basic regions of each monomer in position to embrace the DNA target like a pair of thongs or the cloth pin. Similarly, the bHLH proteins dimerize through helix loop helix motif, which allows the basic part of the long helix to go and bind to the DNA target, and much like the bZip proteins do. And the bHLH as well as the bHLH zip proteins, they bind to DNA in the same way. But then the difference is that the dimerization potential is because of the extra Leucine zippers. So thank you guys for listening to me, and this brings to the end of chapter 9. So I'm so glad. And stay calm, positive, connected, active, nourished, curious, playful, and stay safe. I know it's easier said than done. So thank you guys for listening. And these are the references, voice hugs. So stay tuned until the next.